Hi there, welcome back to our second lecture on the muscular system. You've got me today for this one so you can see my smiling face. We're going to look at the anatomy of a muscle fiber today. Now remember that a muscle fiber is a muscle cell. So whenever I say fiber, I mean cell, and they don't look like what you probably think of with a cell. We already said they're very long. You can see easily here how they're striated, and they have lots and lots of nuclei. For one thing, muscles are made up of a lot of protein. And remember that the nuclei have the DNA for all that protein synthesis, so you need lots of them to constantly be making all these proteins. Muscle fibers are microscopic, but they're really, really long. They run the whole length of the muscle. So if you think about that sartorius muscle, the longest muscle in the body, right, from your ASIS down to your tibia, that long muscle could be 3-4 feet long. Each muscle cell inside that runs the whole length of the muscle. So they're very long and cylinder shaped. Now we have to understand the anatomy of the muscle, and we have to get into this microscopic anatomy so that we can understand how a muscle contracts. So if we're looking at the anatomy of a muscle fiber, you can see this picture here that's in your notes. There's a few key words we have to define. Now, first of all, when we're looking at skeletal muscle, I mentioned before, the prefixes are usually myo or sarc. Sarc means flesh. So notice that we don't have cell membrane. We have a sarcolemma, a sarcoplasm, a sarcoplasmic reticulum. So the sarcolemma is the cell membrane. The sarcoplasm, the cytoplasm, and it has all the organelles that you've learned before, has a lot of mitochondria, has a lot of glycogen in there, stores a lot of glycogen in there, and we have the SR, or sarcoplasmic reticulum. So that's this blue stuff here, and it looks like a network, right? It's the specialized endoplasmic reticulum in muscle, and all muscle has the SR, and it stores calcium. But remember, previously we said that the SR is best developed in skeletal muscle, so skeletal muscle is the best at storing all that calcium. So this is a muscle fiber, right? A muscle cell. So you see there's all kinds of other stuff inside of it. But the blue stuff here is the sarcoplasmic reticulum. Now, if you look at the SR, you're also going to see this little like tube running through it. It's kind of like a lighter color. That's called a transverse tubule. And a lot of time we just call it a T-tubule. So notice that you have this tube right in the middle of the SR. Now, if I go back here for a minute, we have the SR, but then there's something called the terminal cisternae. So this whole thing is SR, but there's areas where it's a little thicker, and that's called the terminal cisternae. So if we look at where the T-tubule is, the terminal cisternae of the SR is usually right next to the T-tubule. So all of the stuff, all of that web-like stuff is sarcoplasmic reticulum, but the thickening part of it right next to the T-tubule, that's going to be called the terminal cisternae. So what we have is a triad, and a triad means it has three structures. So a triad would be each of the terminal cisternaes, so you have them on either side, and the third thing is the T-tubule. So it's kind of like a T-tubule sandwich. You have terminal cisternae, T-tubule. So you might be like, okay, what's going on here? Well, the SR stores and releases calcium. The T-tubule is literally like a tube or a channel that runs, notice it runs perpendicular to the muscle fiber, and that is there for the action potential to shoot through. So the action potential is like electricity, electrical wave, a depolarization wave, and it's going to shoot through that T-tubule, and you can see how if it goes to the T-tubule, it can go all the way across that muscle fiber. And we'll be talking about that when we get into muscle contraction in a later lecture. So here's another view of a muscle fiber, and again in this view you can see the SR, kind of this web-like stuff. In this example, the T-tubules are actually green, so you can see those a little bit better there. I'm going to get another color on my pen maybe. So you can see the green. And again, this is one muscle fiber. Now inside are all these long things, and we saw this previously. Well, they're called myofibrils. So one muscle fiber, inside of it has usually thousands, hundreds to thousands of myofibrils. So myofibrils are cylinder structures. They run the whole length of the cell, and they're made up of protein. Now the muscle proteins are called the myofilaments. So the myofibril is the long tube you can pull out. So they're pulling one out here. And then inside of that is going to be all the muscle proteins. In fact, the reason that muscle looks striated or striped, you can kind of see like a dark and a light color here. So you can see the dark and the light. These are the myosin and the actin, which are the muscle proteins. So the primary muscle proteins are called myosin and actin. Myosin is the thick protein, and actin is the thin protein. So the two main muscle proteins. Put myself over here. So again, muscle fiber made up of myofibrils, hundreds to thousands. Each myofibril is a long protein structure, runs the whole length of the muscle fiber, and it's made up of the proteins. So the myosin is the thick one, so that's gives you like this dark band, and the actin is known as the thin one, and that looks like this lighter area or lighter band that you can see. So the myofilaments are the actual proteins. Okay, so we're going to look at these. Notice, first of all, myosin is much thicker, all right? You can see it's very, very thick, and you can see that actin is thinner, but they're made up of protein subunits, so we need to talk about those. So let's look at actin. Here's actin right here. Now, if you look at actin, I like to compare it if you took a strand of pearls and you twisted it, okay? So imagine twisted strand of pearls. Each pearl, right, each bead, and this example is called G-actin. So notice each of the balls or each of the pearls is called G-actin. And it has a little active site on it. It's a little myosin binding site. So a lot of times we just call it an active site. That's what myosin wants to grab for muscle contraction. Now, F-actin would be all the pearls together on the string. So each pearl would be a G-actin. I think of F-actin as final actin, so they're all put together. But notice there's some other proteins here. So the other proteins that we see are this like blue thing here and this green one. Excuse me. So that's called troponin and tropomyosin. And we usually just refer to this as the TT complex. Okay, so troponin is kind of like shorter. You see it's kind of short and thick. And tropomyosin is really long and skinny and thread-like. And so the two hook together. And what they do is they form this TT complex. And notice that they cover up. See those little black circles? And over here you can see those binding sites. Remember those little active sites? They cover them up. So I call that the cellular chastity belt. So the TT complex is the cellular chastity belt. What it's doing is it's physically blocking, it's covering up that binding site so myosin can't get to it. So if we're going to have muscle contraction, myosin's going to grab the actin and pull it. Well, it can't do that as long as that TT complex is in place. So we're going to need a key to the chastity belt, and we'll get there soon. Let's look at myosin. It's the thick protein. It's actually made up of these club head things. So you see these two heads called club heads with a long skinny tail. So there's a whole bunch of these together. Notice all the tails kind of get assembled in a bundle, and then you have all the club heads. Well, those club heads hinge. Okay, so you have the tail, and then the head can swivel and hinge. So when it grabs actin, it can pull it, which will be important when we look at muscle contraction. Now, by the way, when it grabs actin, it forms something called a cross bridge. So cross bridge means that myosin has found actin and it's grabbed it. Now, there's many, many proteins, by the way. I'm just covering two other ones. There's one called titin and dystrophin. So both of these are more like structural proteins. They're not functional, meaning they make the muscle contract. So titin helps anchor the myosin and helps with the muscle being elastic. Remember, that helps with recoil. And dystrophin or dystrophin is another supporting protein. It was really hard to find a good picture of it, but it actually helps to anchor basically the muscle, the myofibrils to the endomysium and to the muscle. So it helps with the support and the structure of the muscle and helps anchor it together. So if we were to take a muscle fiber, a muscle cell, inside of it are all these myofibrils. So I'll go back to this picture. Remember, we have myofibrils. If we pull one myofibril out, we see these repeating units. You see here you kind of see this repeating thing over and over and over. That's called a sarcomere, and that's the level we have to go to for muscle contraction. So each myofibril, long protein strand, is made up of repeating units called a sarcomere. Again, you're seeing sarc again for flesh. So the sarcomere is the basic contractile unit of muscle. This is where the muscle contraction occurs. And if you look, you can see the sarcomere in the histology, and we'll do the drawing in our notes first. So what we notice is we have this thick and thin pattern because we have the thick protein, which is all the myosin, and then we have the thin, which we're going to see is the actin. So you can see this on a histology slide. So the anatomy of a sarcomere, what do we have going on if we're doing one sarcomere? Well, the boundaries of the sarcomere are made up of this zigzag line. You can see here and you can see here. So I don't care if you call it the Z line or the Z disc. Doesn't matter to me. So that's the ends. That's the boundary of each sarcomere. So the sarcomeres repeat. So they run from Z line to Z line. Now, if we look, we have myosin and actin. The area that contains all of the myosin, so I'll draw the line here. Notice you can see the myosin from one end to the next. That's called the A band. So the A band contains, think A for all. Okay, all of the myosin is there. I'm going to get another color on my marker here. Now the I band, on the other hand, is going to contain all of the actin. So we're going to do the I, well, that was supposed to be a lighter color. Let me try this again. Having some difficulties with my pen. I'll draw it in white. There it goes. So we have the I band is the actin. Okay. And it's this whole structure would be the I band. It goes on either side of the Z line. Now we have an H zone and an M line. So first of all, when we're looking at the A band, remember it's all of the actin, and then all of the myosin. And so one end to the next. And then notice that the actin, I switched colors here so you can see it a little bit better. Notice there's some actin, this kind of like blue stuff, where it crosses over the myosin. So we're in the A band right here. But inside the A band is this area called the zone of overlap. So what that means is there's an area in the A band where the myosin and the actin overlap a little bit. So that zone of overlap is just that region of the A band that you can see. Okay. Now there's an H zone and an M line. The H zone only contains myosin. So that's going to run kind of right up between the two. So I'm going to get another color here. Let's see if I can find something. I'm going to use the obnoxious green again. So right here is the H zone. So I'm doing the green because notice that only contains myosin. And then the M line is just a line that runs right up the middle of the sarcomere. And they don't even know exactly what the M line actually does, but it comes right up the center of the sarcomere. So if we were to draw and label a sarcomere, here's another picture, we can do this because again, we know that the A band, remember the A band is all of the myosin, right? So it's that whole dark thing. You can see the H band, right, in the middle. You have some overlap where the actin overlaps because the H band's only going to be myosin. You can see the Z line, right, the little zigzag line there. Use yellow here. And then the I band is all of the actin. So all of the light area is always going to be just the actin. So we can actually look at the sarcomere and kind of understand what's going on if we were to do like cross sections through it. So if for some reason we took a cross section and we did it right through the H zone, notice that it's only thick filaments because it's only myosin there. But if you did a cross section through the zone of overlap, do you see how you have thick and thin? Because that has myosin and actin. And if you did a cross section just through the I band, you would only have thin because that's only going to be the actin. So that's the anatomy of the sarcomere. And so what happens is, if we look at another picture now, let's try to bring this back. We looked at a muscle. We know that inside the muscle are these bundles of muscle fibers called fascicles. If we pull one muscle fiber out, that's made up of myofibrils. If we were to take one myofibril out, that's made up of repeated units called sarcomeres. And sarcomeres are made up of myosin and we see myosin in the A band, which is all of it, and then we have the H zone, which is only the middle part, which is only the myosin, and then we always see the actin as the lighter area because that's in the I band. And we have to understand this because what happens is when muscle contraction occurs, the actin is going to slide towards the H zone. In fact, the actin, or we can say the I band, so all of that actin moves toward that H zone. So you can see here, if we look at our picture, there's the H zone, right? Just the myosin. It basically disappears because what happens is the myosin pulls the actin and it just shortens. Now the myosin and the actin don't shorten. The sarcomere shortens, kind of like an accordion. It just kind of comes closer together. And when you have hundreds of thousands of them all coming together, then that's going to cause the whole muscle to contract. So the myosin cross bridges have to get a hold of actin. They have to grab a hold of that active site. So we have to get that TT complex out of the way. And when it happens, they just kind of pull like oars on a boat or tug of war, and they just slowly pull that together, and the H zone disappears. And the Z lines, you can see how this gets closer together now, right? It's further apart here. And then that's going to create the tension, which is going to cause the muscle contraction. Now, we know that skeletal muscle is under control of your central nervous system. And so we know that you must have a stimulus from a motor neuron. So you absolutely, for skeletal muscle, remember, nerves are made up of neurons. Those neurons are going to go out and they're going to excite that muscle. If you cut that nerve or that neuron, muscle is paralyzed. It can't contract. So this is an overview of the anatomy of skeletal muscle, of the muscle fiber. And in our next lecture, we're going to talk all about the physiology of how it contracts.